name is Micah, and I'm here with four University of California students
Hello.
who will be helping me explain my research throughout this lecture. We're going to talk about the aspects of learning and how people process information through the nervous system. I combine different subjects like chemistry, anatomy, and neuroscience to explain how the brain works in response to learning and storing short-term and long-term memory. Before I specifically talk about learning, me and several college students will describe the nervous system and its chemical properties, all in which contribute to all kinds of learning. After that, I'll show you different factors that may have an influence in your learning abilities. I feel the last part.
This is an example.
Part 1. The neuron and its electrical properties. The building blocks of the nervous system are called the neurons, or nerve cells. Neurons are highly specialized cells that even today is only partially understood. It was first viewed by a physician named Camillo Golgi, who developed a way to stain neurons, chemically darkening them so that their finer details stood out under a microscope. His stained samples of nervous tissue showed a web of neurons, individual nerve cells that come close to each other but never quite touched.
The neuron contains a nucleus and other parts common to cells, but the neuron is so unique because in complex animals, it ceases to divide or reproduce after a certain point. In humans and other mammals, that point is reached at birth. Although connections among neurons are formed throughout life in these animals, no new nerve cells are produced.
Neurons are also different from other cells because of their well-developed ability to send messages through a combination of electrical and chemical means. The nerve cell is able to do so because of its specialized structures. Sprouting from the cell body are string-like processes called dendrites and one long, relatively thick extension called the axon. These two structures led Ramon E. Cajal to describe the nerve cell as the aristocrat among the structures of the body, with its giant arms stretching out like the tentacles of an octopus to the provinces on the frontier of the outside world to watch for the constant ambushes of physical and chemical forces.
Dendrites receive signals from neighboring neurons and conduct these signals back to their own cell, whereas the axon conducts the impulses away from the neuron to other nerve cells. The axon can run up to several feet in length and contains a number of branches that allow it to make connections with many other cells as well. The point at which it makes contact, the space between the axon and its target cell, is a gap called the synapse, across which one neuron transmits information to another.
Nerve action was first discovered to be associated with electricity in the 1780s by the Italian anatomist Luigi Galvani, who showed that an electrical charge made muscles contract. In one of his experiments, Galvani attached a lightning rod to a severed frog leg, stretched a wire from the leg to water, and waited for an oncoming thunderstorm. When lightning flashed, electrical impulses carried by the frog's neurons caused its muscles to contract frequently and intensely.
In the late 19th century, the German chemist and physicist Walter Hermann Nernst proposed that the electrical activity of the neuron results from the passage of ions, or electrically charged atoms, through the cell's outer membrane. Finally, in the early 1950s, British scientists Alan Lloyd Hodgkin and Andrew Fielding Huxley, through experiments using the giant axon of the squid, were able to work out the step-by-step sequence leading to the conduction of an electrical impulse from a neuron.
So as you can see, we have some guest speakers from UCLA, UC Irvine, who will be helping us with our academic lecture about the nervous system. Our first guest speaker here is Visat Lim, a chem major in UCLA, who will be talking more about the electrical impulse and the chemical transmission of the neuron.
Basically, there's a lot of chemistry involved when neurons transmit information from one neuron to another. Ions are found in the fluids that bathe the body's cells. Chief among these atoms are potassium and sodium ions, which are equally and positively charged. The membrane of a neuron, like that of other cells contains many holes called ion channels, which allow specific kinds of ions to pass in and out. Some channels, for example, will only let potassium ions through, whereas others can only be crossed by sodium ions. In addition, some of the channels for these ions have gates that control when an ion can go in or out. Other channels are always open. Most potassium channels do not have gates, thus potassium ions can freely pass through the membrane at any time. Most sodium channels have gates that are open on some occasions and close on others. When at rest, a neuron contains a high concentration of potassium ions and a low concentration of sodium ions compared to the fluid that surrounds and bathes it. Thanks to a mechanism that pumps sodium out of the cell and potassium into it. Even though potassium and sodium ions are positively charged, the neuron has a negative charge relative to the fluid outside it because the nerve cell also contains negatively charged protein molecules. Because of their large size, these protein molecules are unable to pass through any ion channel in the membrane and so are trapped inside the neuron. When a nerve cell is stimulated, say by plunging one's hand into a tub of warm water, sodium channels open suddenly. Sodium pours in partially because of the electrical attraction of the positive sodium to the cell's negative charge. The inside of the cell then becomes positively charged, which as a result sends an electrical impulse sweeping down the axon until this signal reaches the synapse. Once the neuron has fired, sent out an electrical impulse, the normal balance of ions is restored and the cell stops discharging electricity. Of course, the sensations people feel often last much longer than the split second of an impulse firing. For example, the hand soaking in a tub of warm water will continue to feel warm until it is lifted out again, because the nerve cells continue their cycle again and again, firing an impulse, restoring their normal ion balance, charging up again, firing another impulse, and so on. To describe these events, scientists use the language of physics, when the ion balance is normal and the neuron is not being charged. The resting nerve cell is said to be polarized and to have a resting potential, or the negative charge relative to the outside of the cell. When the neuron gears up to fire, the membrane depolarizes. The electrical impulse then sent is called the action potential. Bing bing.
Yay! Okay, I don't know if there's more. What was that?
Go on.
Hi, I'm back. So, the speed with which an impulse travels down a human nerve fiber depends in part on the diameter of the fiber. The larger the diameter, the faster the impulse. The thinnest fibers conduct impulses at a speed of about 40 inches per second, or roughly 2 miles per hour. The thickest fibers conduct about 100 times faster. The speed of an impulse also depends on whether the fiber is insulated. On some nerve cells, a fatty substance called myelin forms an insulating sheet around the axon. This allows the neuron to conduct an electrical impulse more efficiently and therefore more quickly. When the myelin sheet is damaged, as in a disease called multiple sclerosis, nerve cell function is severely impaired.
Whatever the speed of the impulse, its magnitude is always the same. A strong impulse, such as stepping on a tack barefoot, can make a nerve cell fire more frequently, but it does not make each firing stronger. However, the strength of the stimulus determines whether a nerve cell does or does not fire. When the stimulation is strong enough, the neuron's membrane reaches what is called its threshold potential and the nerve cell fires. The changes that cause it to fire are called the all or none response because they either occur or do not occur depending on the strength of the stimulus.
Oh, alright, thank you, Vislav. Next up, we have a mechanical engineering student named Tiraniti Samsuk from UC Irvine who will expand more about the chemical transmission of the neuron.
Cut that out.
Running down the axon, the electrical impulses reach the synapse. For a long time, there was a rigorous debate about how the nerve cell transmitted the information across the gap. However, before the electrical nature of the neuron was clarified, the transmission debate was settled. In 1921, a German physiologist, Otto Lewey, determined that a chemical called acetylcholine transmitted signals across the synapse from one nerve cell to another. As it turns out, acetylcholine is only one of the many such signal transmitting substances called neurotransmitters. When, in, when an electrical impulse reaches the synapse, neurotransmitter molecules are released from the end of the axon. They then cross the gap and act on the adjacent neuron. These molecules may activate the neuron so that the electrical signal continues its journey. In other cases, however, the impulses tell the nerve cell not to pass them to another neuron. The neurotransmitters are then either absorbed by the neuron that released them or swept them away by what we are now called scavenger molecules. Research in recent years has revived the idea that in rare cases, signals can be transmitted from nerve to nerve by electrical impulse alone, with no help from neurotransmitter chemicals. It is true that neurotransmitters do the job at most at synapse, but there are some places in the nervous system where electrical signals jump directly from neuron to neuron. This kind of transmission may exist where a speedy response is of the essence.
Okay, thank you, T. Now more about the organization of neurons. It is rare for a nerve cell to make contact with only one other neuron. As mentioned, a neuron usually sends out its axon to form connections with many other cells. In addition, an average-sized neuron in the spinal cord has hundreds of dendrites. Thus, it can simultaneously receive hundreds of signals from other neurons, some activating the nerve cell so that it will pick up and send an electrical impulse from other neurons, and some preventing it from passing on signals. This sort of complex interaction goes on constantly. Neurons are found in groups with their axons bunched together like stalks. In the central nervous system, the brain and spinal cord, these bundles of axons are whitish in color and so are called white matter. The main part of each cell, the cell body where the nucleus resides, is grayish in color. Thus, a mass of cell bodies is called gray matter. In the peripheral nervous system, the network of neurons outside the central nervous system, collections of cell bodies are called ganglia. And bundles of axons are called neurons, as opposed to nerve cells, which consist of the cell bodies and axons together.
Neurons do not act in isolation. They almost invariably come with companion cells called neuroglia that help them function. In the central nervous system, there are four kinds of neuroglia: astrocytes, ependymal cells, microglia, and oligodendrocytes. each type offering some sort of protection for the neurons, from fighting infection to forming a barrier against harmful substances in the bloodstream. Each has its own unique structure and function. In the peripheral nervous system, there is only one kind of neuroglial cell, the Schwann cell, named after German physiologist Theodor Schwann. Schwann cells not only manufacture myelin, but also appear to play an important role in the repair of nervous tissue. This function dramatically distinguishes the neurons of the peripheral nervous system from those in the brain and spinal cord. When a neuron in the spinal cord is severely injured or cut, it does not regenerate. Neurons in the peripheral system do regenerate, however.
You really have to add that.
Part 2, Learning and Memory.
Of all the mind's capabilities, learning and memory may be the most remarkable. Memories can be revived after being locked up for a decade, and the store of them seems inexhaustible. The researchers have been studying the brain for centuries, but only within the past decade or so have they begun to understand the biological mechanisms of learning and memory.
The best information on the nature of the physical changes that create memories comes from animal experiments. In these, scientists have looked more or less directly at neurons before and after learning takes place. The best known series of experiments has been done during the last 30 years by Eric Kandel at Columbia University. Kandel works with the California sea-dwelling snail, Aplysia californica, whose entire nervous system contains only 20,000 neurons. However, these neurons are large, which allows Kandel to measure what happens within them during the learning process. Aplysia has been found to have the ability to learn. If touched gently on its siphon, a breathing organ on top of its gill, Aplysia retracts the gill slowly. If it is alerted to the possibility of danger, if, say, electric shocks to its tail, the snail quickly covers the siphon and gill with a protective flap of skin. Sensory and motor nerve skills link the tail and the siphon. When the tail is shocked a number of times, the sensory neurons, the nerve cells at the siphon at the end of that link, accumulate an unusually large amount of neurotransmitter molecules. Another shock releases these molecules, giving a sudden, strong message to the motor neurons that act on the muscle that pulls the siphon and gill under the protective flap. This is a good model of sensitization, a kind of learning that puts the nervous system on alert. For example, because experience has taught some circuits in the human nervous system that the sound of an automobile horn means danger, a person crossing the street may be startled upon hearing such a sound. There is another kind of learning called habituation, which occurs on repeated exposure to a not very meaningful stimulus. If, for example, an automobile horn were to go off every 5 minutes in the street outside a person's room, that person would soon stop paying attention to the sound. In Aplysia, Kandel produces habituation by repeated gentle squirts of water on the siphon. After a while, the snail does not respond to the stimulus, because the neurons in the circuit do not release enough neurotransmitter to cause the motor neurons to fire. Thus, a change in the amount of neurotransmitter in nerve endings is one mechanism for learning. Now let's talk about increasing neural connections. A change in the number of contacts between neurons is another mechanism of learning. Neuroscientists have seen such changes in several experiments. At Columbia, for example, Craig Bailey and Mary Chen have shown that as learning takes place, there is an increase in the number of dendrites that the sensory neurons of Aplysia's siphon sends out to make contact with the motor neuron, as well as an increase in the neurotransmitter contact at the synapses. In experiments reported in 1989, Richard Thompson of the University of Southern California and William Greenough of the University of Illinois detected similar changes in rabbits. Thompson and Greenough trained the rabbits in Pavlovian fashion, sounding a tone while blowing a puff of air against the rabbit's eyes. After a while, the rabbits would blink their eyes whenever the tone was sounded. Careful studies of brain tissue in the trained animals showed increased numbers of dendrites. The researchers concluded that the cerebellum is essential for learning such conditioned responses. We don't yet know all the details of how the brain reprograms itself, but it has become clear that, as Freud proposed, the secret lies mainly in the rich chemical broth of our synapses. What goes on in the microscopic spaces between our neurons is exceedingly complicated, but in simple terms, it involves various chemical reactions that register and record experiences in neural pathways. Every time we perform a task or experience a sensation, whether physical or mental, a set of neurons in our brains is activated. If they're in proximity, those neurons join together through the exchange of synaptic neurotransmitters, like the amino and the glutamate. As the same experience is repeated, the synaptic links between the neurons grow stronger and more plentiful through both physiological. As the same experience is repeated, the synaptic links between the neurons grow stronger and more plentiful through both physiological changes, such as the release of higher concentrations of neurotransmitters, and anatomical ones, such as the generation of new neurons or the growth of new synaptic terminals on existing axons and dendrites. Synaptic links can also weaken in response to experiences, again as a result of physiological and anatomical alterations. What we learn as we live is embedded in the ever-changing cellular connections inside our heads. The chains of linked neurons from our minds, true vital paths. Today, scientists sum up the essential dynamic of neuroplasticity with a saying known as Hebb's rule. Cells that fire together, wire together. Even if neurons rarely reproduce after a certain period, they still continue to rearrange themselves in different ways based on what we're exposed to. Therefore, the brain is not the rigid processing machine we once thought it to be. Though different regions are associated with different mental functions, the cellular components do not form permanent structures or play rigid roles. They're flexible. They change with experience, circumstance, and need. Some of the most extensive and remarkable changes take place in response to damage in the nervous system. Experiments show, for instance, that if a person is struck blind, the part of the brain that has become dedicated to processing visual stimuli, the visual cortex, does not just go dark. It is quickly taken over by circuits used for audio processing. And if the person learns to read Braille, the visual cortex will be redeployed for processing information delivered through the sense of touch. Neurons seem to want to receive input. When their usual input disappears, they start responding to the next best thing. Thanks to the ready adaptability of neurons, the senses of hearing and touch can grow sharper to mitigate the effects of the loss of sight. Similar alterations happen in the brains of people who go deaf. Their other senses strengthen to help make up for the loss of hearing. The area in the brain that processes peripheral vision, for example, grows larger, enabling them to see what they once would have heard.
Plasticity is a normal ongoing state of nervous system throughout the lifespan. Our brains are constantly changing in response to our experiences and our behavior, reworking their circuitry with each sensory input, motor act, association, reward signal, action plan, or shift of awareness. Neuroplasticity is one of the most important products of evolution, a trait that enables the nervous system to escape the restrictions of its own genome and thus adapt to environmental pressures, physiologic changes, and experiences. Natural selection has not designed a brain that consists of numerous prefabricated adaptations, but rather one that is able to adapt to local environmental demands throughout the lifetime of an individual, and sometimes within a period of days by forming specialized structures to deal with those demands. Evolution has given us a brain that can literally change its mind over and over again.
Our ways of thinking and perceiving and acting, we now know, are not entirely determined by our genes, nor are they entirely determined by our childhood experiences. We change them through the way we live and through the tools we use.
And that's how we get scarred by exams
Okay.
and classes like AP Chemistry.
Part 3. How does food, sleep, and stress affect learning?
Sleep is considered an important aspect of learning and memory, both early in development and throughout life. Research on learning and memory again focuses on REM sleep. REM sleep is a stage during which brain waves are slow and the sleeper experiences rapid eye movement and irregular heart rate and respiration. It is the only stage of sleep in which dreams occur. Animal studies find that deprivation of REM sleep slows the learning process, and conversely, during periods of intense training, REM sleep increases. One theory based on these studies states that knowledge gathered during waking hours is stored in permanent memory during sleep by changes in brain cells, which form new connections for new memories. Research suggests that sleep helps learning and memory in two distinct ways. First, a sleep-deprived person cannot focus attention optimally and therefore cannot learn efficiently. Second, sleep itself has a role in the consolidation of memory, which is essential for learning new information. Although the exact mechanisms aren't known, learning and memory are often described in terms of three functions. Acquisition refers to the introduction of new information to the brain. Consolidation represents the processes by which a memory becomes stable. Recall refers to the ability to access the information, whether consciously or unconsciously, after it has been stored. Each of these steps is necessary for proper memory function. Acquisition and recall occur only during wakefulness, but research suggests that memory consolidation takes place during sleep through the strengthening of the neural connections that form our memories. Although there is no consensus about how sleep makes this process possible, many researchers think that specific characteristics of brainwaves during different stages of sleep are associated with the formation of particular types of memory.
Alright, so now we're going to talk about food. Yay! Are you serious?
Go. Food can have a big influence in your mood and learning abilities. If you are an evening person and you skip breakfast, your cognitive performance might be impaired.
That's my line.
A large breakfast rich in protein, however, could improve your recall performance but might impair your concentration. This illustrates the complexity of relationships between food, mood, learning, and the need to find a healthy balance of food choices.
It's your line.
That's
You
your
stole
line.
my lines. Just go.
Now you read my line.
Okay. So, food is like a pharmaceutical compound that affects the brain. Diet, exercise, and sleep have the potential to alter our brain health and mental function. This raises the exciting possibility that changes in diet are a viable strategy for enhancing cognitive abilities. Omega-3 fatty acids found in salmon, walnuts, and kiwi fruit provide many benefits, including improving learning and memory and helping to fight against such mental disorders as depression and mood disorders, schizophrenia, and dementia. Omega-3 fatty acids support synaptic plasticity and seem to positively affect the expression of several molecules related to learning and memory that are found on synapses. A deficiency of omega-3 fatty acids in rodents results in impaired learning and memory.
Emerging research indicates that the effects of diet on the brain, combined with the effects of exercise and a good night's sleep, can strengthen synapses and provide other cognitive benefits.
Okay. Stress is another factor that may affect learning. Although it is known that long-term or chronic stress can affect the brain's learning and memory region, a new finding discovers short-term stress, lasting as little as a few hours, can also impair brain cell communication in these critical areas.
Stress is a constant in our lives and cannot be avoided. Yep, totally. Our findings can play an important role in the current development of drugs that might prevent these undesirable effects and offer insights into why some people are forgetful or have difficulty retaining information during stressful situations. In their study, Dr. Tali Baram and her UC Irvine colleagues identified a novel process by which stress caused these effects. They found that rather than involving the widely known stress hormone cortisol, which circulates throughout the body, acute stress activated selective molecules called corticotropin, releasing hormones which disrupted the process by which the brain collects and stores memories.
In rat and mouse studies, Baram's group saw that the release of CRH in the hippocampus, the brain's primary learning and memory center, led to the rapid disintegration of dendrites, which in turn limited the ability of synapses to collect and store memories. The researchers discovered that blocking the CRH molecule's interaction with their receptor molecules eliminated stress damage to dendritic spines in the hippocampal cells involved in learning and memory.
You know what? I'll restart that. In addition, the authors replicated the effects of stress on dendritic spines by administering low levels of synthetic CRH and watching how the spines retracted over minutes. Fortunately, once we removed the CRH, the spines seemed to grow back, Baram said. Baram also noted that there are compounds under development that show the ability to block CRH receptors, and that this study can play a role in the creation of therapies based on these compounds to address stress-related learning and memory loss. Ah, that's pretty cool.
Both early in development.
Running down the axon, the electric. I hate reading.
That surrounds and baits it.
Oh.
Okay. Continue. You're just gonna cut it.
Yeah. Wow. Okay. Just wave it. Oh, your pinkies. Mine is so small.
Oh, whatever.
Chloe, show it. Like,
I mean, my hands are a lot bigger than yours, so.
no, but it's relatively longer than.
Don't forget this time.
Finish, hurry up.
Plasticity is a normal, ongoing
Dude,
state of the
that
nervous
was
system throughout the
You're
lifespan.
like,
Our
brains
brains are constantly changing in response to our experiences and our behavior.
Restart! With more energy!
Circuitry with each sensory input, motor act, association, rewards, and action. Awareness. Neuroplasticity is one of the most important parts of evolutions. A trait that no one's your name. Hello, class. That is so creepy.
You'd be the creepiest professor ever.
Hello, class. Today I will be teaching you about plasticity. Plasticity is the normal ongoing state of the nervous system throughout the lifespan. Our brains are constantly changing in response to our experiences and our behavior, reworking their circuitry with each sensory input, motor act, association, reward signal, action plan, or shift of awareness.
Wasting my
Neuroplasticity is
time,
one
man.
of the most important products of evolution, a trait that enables the nervous system to escape the restrictions of its own genome. How do you know? Genome, right? Yeah.
Yep.
Genome. That's genome. That sounds weird. Genome, right? Oh, genome. That sounds weird.
Genome?
Genome. That
You're a
sounds so
biochem
weird.
major.
I'm a chem major.
You're a biochem major.
I'm a chem major.
Don't be like him.
I'm a chem major. More specifically, I'm a physical chem. So. Psh.
Oh my.
Protein molecules. I'll just repeat the whole paragraph. Gosh, what do you want from me?
You need more energy. You
Hi,
like you're
guys.
you look like you're you look like you're just reading up or something, which you are, but like.
Yeah, because you didn't give me time to prepare anything.
Okay,
you
fine.
basically.
Then, okay, fine. Then read the whole thing.
No. You basically just come and say, okay, read all of this. So, do you want me to like magically know what is on the thingy?
Yeah.
Okay.
You know that you're smart, but stop.
Yeah, but it's not like I've learned these things before.
Okay, how about you read it first?
No, it's okay.
And
Why do I always mess it up? I have to read it first.
Dude, that's exciting. Don't have weird friends in common.
You're all weird. When a nerve cell is stimulated, say by plunging one's hand into a tub of warm water.
Plunging.
Okay, sodium channels open suddenly. Sodium pours in partially. Because of the electrical attraction of potassium sodium to the cell's negative charge. The inside of the cell. You're not putting this into the video, are you?
Maybe. Watch it gonna be in the video.
Oh my gosh.
Say hi to Falk.
Yeah, hi, Mr. Falk.
Okay, you're going.
When a nerve. When a nerve.
Oh my gosh. Just read the thing out. How hard is that?
Okay, you read it.
When a nerve cell... Hurry up, I'm tired.
Gosh, so demanding.
Alright, so now we're going to talk about food. Yay! Now we're going to talk about food. Yay! Come on! Okay. Supposed to say yay. Okay.
Hey, that was cool. You should just add that. One is excited and one is like really.
Now we're going to talk about food. Yay! That's not what I meant by that. Oh my. I'm sorry. No, just don't. Just don't.
Wait, I'm trying to find my evil laugh. That sounds lame. Hurry up, let's go.
That's how weird we are. Sorry. Those are highly specialized cells that even. Oh wait, we need a.
Neurons are highly specialized cells that even today is only partially understood, like quantum mechanics. It was that.
Brother's so weird.
We're too tired.
That's what midterms do to you. So, have fun in college. More energy, dude.
I don't have any.
Sheesh.
Don't you see I'm relaxing right now? I could be studying, but I don't have enough energy to go on.
You're just watching something.
Yeah.
Hello, students. My name is Micah.
My name is Micah.
40 inches per second, or roughly 2 miles per hour. The thickest fibers conduct about 100 times faster. The speed of an impulse also. What? What?
Just thought of something funny.
What? There's a 40 inches. Huh?
Restart it. This
Well,
is
I'm
fun. I'm
thinking of-
wasting your time.
Thinking of something. What is funny? The speed of light.
There's nothing funny about that. Trust me.
Hi, I am back. Okay. So, the speed with which an impulse travels down the human nerve fiber depends in part of the diameter of the fiber. The larger the diameter. Okay, kind of. Kind of.
I don't know what's wrong with her.
Yeah, you don't know what I'm talking about. Read it then.
Ependymol? Is
Mhm.
ependymol soil? How did I read cells into soil?
What?
Ependymol cells.
Ependymal cells.
I read cells. Microglia and oligodendrocytes.
You don't know those words?
Huh?
You never seen those words before?
No, actually not.
Like in bio. Oh no, I think
No, I
it
wasn't in bio. No. I don't think it's not
Yeah,
in
well,
bio.
you think
Because we don't even go into nervous system. Actually, I don't even remember anything from AP bio. I don't even
You
know
got a
how I passed
4.
AP bio.
you got a 4.
To be honest, I don't even know how I got that.
got a 5 on a practice exam. Yeah, you got it. Okay.
Hey, don't touch my hand. I'm older.
No. I want to grab your brain. Oh!
Glial. Neuroglial. Why do you never tell me there are like these surprises that I should know first ahead of time? How many times do you have to tell you, Michael? Did you have to inform me about these kinds of words
It's
first?
just reading. You're supposed to know.
The Schwann cell?
Yeah. Wait. Start
The-
over the sentence. The sentence.
The Schwann cell. The Schwanns. A Gebrüche de Schwanns. Never the Schwann. Are you sure it's Schwann?
Start over.
I don't think it's called Schwann. Because it's a
Schwann.
German
It's
name.
Schwann.
It's a German
I've
name.
heard of it. I heard
Really?
my kids said it. I heard Jay
Okay,
said it.
the Schwann cell name.
And I think it's neuroglia, not neuroglia. I'm pretty sure.
Neurons. Neurology. Neurology.
Neuroglia.
Neuroglia.
Neuro.
Neurosurgeon. Neuroglia.
Okay, I guess it's neuroglia.
I guess neuroglia also works.
Okay.
I guess neuroglia.
Alright, start from here.
Is it neuroglial? Neuroglial
Neuroglia. Choose one.
No, neuroglial cell or neuroglial cell? Glial?
Neuroglial. It's
Glial?
Glial.
Glial? No, it's an action. Neuroglial or neuroglias?
Neuroglial.
Neuroglial.
Just start!
I love it. It does not regenerate. Neurons in the peripheral system do regenerate, however. However.
Really have to add that.
However.
Oh my gosh.
Neurons do not act in isolation. They almost invariably 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 Neurons do not act in isolation. They almost invariably come with companion cells called neuroglia. Neuroglia?
Neuroglia.
Because there's a song that is called Pasaglia.
What's that song?
Probably Pasaglia. Neuroglia? Is it?
Yeah.
Or
Start
neuroglia?
over.
Is it neuroglia?
Neuroglia.
Are you sure? Not glia.
Okay, let's get up.
Neurons do not act in isolation. They almost come invariably. Shush.
Okay.
Neurons. Corticotropin.
Start from here.
They found rather than the involving the widely
With
known stress
energy.
hormone cortisol,
We
which
start
circulates.
with
Oh my
energy.
god! Start with energy. Is that how you do it?
Yes.
Oh my god! Wait, is it recording?
Yeah.
Oh yeah, it is. Okay.
Okay.
They found rather than the. They found that rather than involving the widely known stress hormone